Good day, everyone. And sorry, there was a little bit delay. And I guess now we are, how many? I can see how many we are. So just short of 60 people, so we can start the workshop of the symposium. So today we are going, me and my colleague, Cornelia Wetzke. So Connie is waving. We are going to showcase label-free FLIM experiments. So the aim of this workshop is that to demonstrate how we can study the cellular metabolism using NADH or NADPH autofluorescence. We have not limited ourselves to only that's NADH or NADPH. We have done the test with FAD. And so far in the last few days, we have talked a lot about metabolic FLIM. We had several talks and long discussions. And since today morning, we have had two workshops talking about Leica SP8 FALCON system. So today we are going to use the same system. For sure, we talk about that in details. However, just going through the system, talking about how the instrument is working, but to make it like more realistic, we have thought about like showcasing one kind of examples. So as an overview, I can go to the next page. Yes. So today that we are going to talk about the role of NADH, NADPH, and FAD as well in the cellular metabolism. And then we showcase about one specific sample. So today that's we are going to present some data, so some kind of measurements we did with Connie on freshly dissected salivary glands of the Drosophila. So she will mainly explain the sample preparation. Then we talk about the instrumentation and review some parameters which are left from the other workshops. Not so many left, we'll see. How to properly acquire FLIM data, and later on how to analyze them. So the analysis today would be mainly focused on the reconvolution and fitting the curves, decay curves. Okay. However, that's we need to know that why it is important for us to assess the cellular metabolism. So many people know the answer, so especially that the people with the background in biology just review very few small cases. That's also that in the talks of Angelika Hook and Amazi Priyasamy specifically, they have talked about the potential and the abilities of this technique, metabolic FLIM, to assess and address clinical diagnosis or biomedical studies. As well as it's important for many cases that people would like to study, for example, that's different signaling pathways in the cell that might be correlated with the changes in the metabolic state of the cell. So if the metabolism actually shift toward glycolysis, that would indicate or trigger some specific pathways, or the other way around with oxidative phosphorylation. And that's the main reason we would like to have a way to assess cellular metabolism, specifically in tissues. So in the cells, there are other alternatives that they are established methods, and yeah, I would say relatively slow to implement and even export data and analyze them. But when it comes to the tissue or in vivo, that's more tricky. And we would like to address this one using FLIM today. So why do FLIM? Why do fluorescence lifetime? So the advantages of FLIM in general, so just to go through that fast, is that it's highly sensitive to the fluorophore conformation and changes in the environment. There was short discussion regarding the microenvironment of the fluorophore, how that affect the lifetime. Well, if one imagine the Jablonski diagram, So there are so many pathways an excited electron can just decay down. One of them is fluorescence. And just imagine what is the competition that's between the decision between different pathways to decay down. And any changes in the ion concentration, temperature, pH, in the microenvironment, inside the cell, inside your cell culture, or around the tissue, or inside an animal model, would affect the decision which is made that the electron would decay through fluorescence or the other ways. And that changes the decay behavior of the fluorophore. It's a very sensitive tool to address changes in the conformation of the fluorophore, the proteins or enzymes that we are studying, with the changes related to the environment or any changes inside the cell. The other way is that it's independent. In some cases, that's in principle independent. I'm not saying that the decay that we are going to analyze at the end is independent from the concentration, but in principle, lifetime measurement, the lifetime of the fluorophore, it's independent from the concentration that we have inside the cell. So that's very important points that we have to keep it in mind. And so also that's fluorescence lifetime measurements are immune from the excitation or detection heterogeneity, especially in thick samples. And also they're virtually independent to excitation intensity. Okay, questionable, we'll talk about that. So the drawbacks are that it's limited to the correlative studies, whereas several biological controls required. We see that how that should be addressed that when you want to do properly doing FLIM experiments. And also that's careful statistical analysis required due to its iterative fitting algorithms and relatively slow in terms of acquisition. So we have heard about fast FLIM, rapid FLIM, FALCON, that they can be super fast. But we always, as usual, we need to pay attention to so many parameters when we do, in general, microscopy. It's always like a compromise of the speeds, signal to noise, the data that we are acquiring, as well as the resolution, temporal or spatial. Which one is more important? We need to always compromise. And that's why I mentioned that it's relatively slow in terms of acquisition. So that's now I hand it to Connie. She will explain the background of the experiments that we have designed and what we would like to measure. Connie?
Yes, welcome everyone. Ali, could we please swap screens?
Yes,
You just
I can.
stop sharing.
I just stop sharing.
Yes, thank you.
I just make you the. Can you share now? Can you share your screen now?
I'm trying. It has worked before.
Yeah. Yes. Great. You have your screen now.
Do you have my? Yeah, here comes my presentation. You can see it. Okay, so welcome again, everyone, to our workshop. What I would like to introduce now is, on one hand, a bit of the background of cellular metabolism and why FLIM is interesting here, and why particularly the molecules NADH and FAD are of importance here. Then I will go into the sample we used in a little test experiment, and I will introduce this. And later on, I will shortly introduce how we prepare the sample. Okay, so now let's start. Why are NADH and FAD particularly important here in cellular metabolism? And why do we use FLIM to investigate metabolism? So generally, NADH and FAD are the coenzymes in any living cell and tissue you can think of any species. Relevant for metabolism and basically in the process of breaking down glucose through a number of biochemical processes you can shortly see, not shortly, but you can see in this scheme here. And the final goal is, in the end, to provide a driving force to drive an ATP synthase, which is, you can see at the very lower end of the screen in the oxidative phosphorylation section, you want to drive an ATP synthase to produce the fuel of all cells, which is ATP. So coming to a bit more detail, so glucose is being fed into glycolysis. And as you can see here, through a number of individual sequential steps, it is broken down into two pyruvate molecules, which are the product of glycolysis. During these steps, two NAD+ molecules are already being reduced, meaning they take up hydrogen atoms. They're being reduced to NADH. At the same time, two molecules of ATP are being produced. Then this series goes on. Pyruvate is fed into the TCA cycle. And as you can see here in the middle part on the right side, now here FAD becomes involved as well. So both FAD, but also NAD+, several of these molecules are being reduced. And that's kind of the key role of NAD and FAD in these initial steps. They take up hydrogen atoms and they serve as key electron carriers through metabolism. Now during oxidative phosphorylation, so in the red part at the bottom, these reduced coenzymes are being further processed in a series, which is called the electron transport chain. And what happens is they become oxidized, and their hydrogen atoms, they're being separated into protons and electrons. So in order to generate a gradient across the inner mitochondrial membrane, so these enzyme complexes sit in the inner mitochondrial membrane, and this in the end provides the driving force in order to produce sufficient and high levels of ATP. Okay, so that's NADH and FAD are the key coenzymes for this process. At the same time, they're highly abundant in cells and tissues. So NADA in particular is present in the micromolar to millimolar range in tissues, while FAD is present in the lower micromolar range normally. At the same time, and why this is particularly interesting now in the microscopy community, both molecules are intrinsically autofluorescent. That means on the left, you see an NADH molecule in which you see labeled in blue here, the nicotinamide substructure of the molecule with its aromatic ring structure. And this substructure of the molecule is capable to absorb either single photons in the UV range or two photons in the near infrared range in order to be excited for fluorescence. In a similar way, it's true for the FAD molecule with its Flavin substructure. You can see on the right side, here highlighted in green, which also has an aromatic substructure, which can be excited to fluorescence and has very similar excitation and emission characteristics as GFP. So it makes it quite nicely, or it makes it quite well convenient, since GFP are classical filter settings that many microscopic systems bring along. And not only are these two molecules intrinsically autofluorescent, at the same time you can consider them as also intracellular biosensors, which means that that their lifetime is actually sensitive to the immediate microenvironment of the molecules, which means in this case these two molecules are sensitive to potential protein interactions. And as you can imagine, these two molecules, since they're involved in metabolism, the obvious candidates for protein interactions are enzymes of metabolic pathways here. And I will now give you a short overview of how this intrinsic fluorescence and its lifetime is being used, and how this can be interpreted in the biological context. Just as a very simple rule of thumb, what often applies to NADH and FAD FLIM, they tend to behave in the opponent way. So what is true for one molecule often is the opposite for the other one. And you will see what I mean in this slide. So as you've probably recognized before, NADH is fluorescent in its reduced state. As I mentioned, we excited in our measurements using 740 nanometers, and it emits light in the range of 450 nanometers. Regarding its lifetime, If it's in a free, not protein-bound state, then NADH has a relatively short fluorescent lifetime in the range of 400 to 500 picoseconds. However, if it interacts with proteins, with, in this case, is frequently enzymes, then this lifetime is prolonged into the nanosecond range. And this lifetime lengthening, or these long lifetimes, are actually very much dependent on the interacting proteins. Therefore, a certain range is given between 1 and 3 to 4 nanoseconds. There have been, in the past years, nice publications, for example, by the lab of Melissa Scala, showing in vitro experiments how actually the interacting protein affects the NADH lifetime. And I think this is a field which requires a lot more attention, and there's still a lot more to learn and find out about metabolism. Now coming to FAD, on the contrary, it is fluorescent in its oxidized state. And as you see in two-photon microscopy, be excited in the range of 880 to 900 nanometers, and it emits in the lower 500 nanometer range. Its lifetime is also dependent on protein interaction. In this case, it is the short lifetime fraction, which represents the protein or enzyme-bound FAD subfraction. Here, the lifetime is even shorter in the range of 0.3 nanoseconds, while the free FAD has the longer lifetime in the range of 2 nanoseconds. All of this has been nicely introduced also on the first 2 days of the talks by Amasi Periasamy and also by Angelika Rück. But this is just as a small update and reminder also for everyone who had not joined earlier. So now, what is the biological interpretation? How can these measurements be used to understand your biological system? So as I mentioned before, NADH has a long and a short lifetime fraction. Very frequently, it's fitted with a bioexponential decay. And the short lifetime fraction, being the free NADH molecule, is associated with glycolysis, while the protein-bound form with the longer lifetime is mainly associated with the oxidative phosphorylation cascades, or OXPHOS in short. For FAD, this interpretation is much more difficult, and there's some discussion in the field, or different labs see different tendency of how this can be interpreted. Often this is very much cell type or tissue specific, possibly due to different also presence of FAD levels and other Flavin containing molecules. But generally, again, the unbound form has the long lifetime, the protein-bound form has the short lifetime. As you all know, the mean lifetime is calculated, or is the sum of the individual lifetimes as a product with the relative fractions, which mean that if you only consider the mean lifetime, you may lose additional information which is part of these either individual lifetime components or the relative fractions. So when you do lifetime analysis in this context, it's always interesting to consider all of these parameters being the short and the long lifetime per se, but also the relative fractions for analysis. And we will also focus mainly on the percentage of the short NADPH lifetime fraction. And mentioning this again, the higher this is, the more free NADH, the more glycolytic is the cell or tissue. Often this is being used also in the context of stem cell biology or tumor biology. So the more glycolytic, a more stem cell state is present in cells or tissues, or many of the tumors become more glycolytic as upon progression. And the inverse is the case in a more oxidative state. Then this data can indicate that your study specimen is either more differentiated or less tumorigenic. It's kind of a general interpretation that is often applied. But of course, this all depends on your special system, model system you're investigating. And in the past 2 years, the lab of Amasi Periasamy has introduced the so-called FLIM redox ratio, in short FLIR. And this is now a lifetime-based redox ratio, which can be calculated exactly using 2 of these parameters from the analysis. So previously, the redox ratio has been intensively used, but these measurements have been done on the basis of intensity measurements, meaning that you compare the intensities or ratios of NADH fluorescence and FAD fluorescence. And here, actually, the relative lifetime proportions of both bound forms of NADH and of FAD are being compared. The interpretation here is also still discussed in the field, and maybe also due to the contribution of FAD, maybe very much depending on your model species. But the group of Amasi Periasamy has found that with increasing FLIR in one of their examples, this increases also in the oxidative state. Okay, so now we'll lead over to a little experiment, which we also use to show you the measurements we've performed also on the system later on. And for this, I will start with a short introductory movie, which introduces shortly our model organism, and then I'll go over shortly to the sample preparation.
Metabolic imaging can be performed in literally any tissue of the organism of your interest, since it is based on the metabolic coenzymes, which are ubiquitous, NADH, and if you like, FAD. For this example,
Connie, this does not seem to work very well. When you shared the screen, did you select the transmit computer audio?
You can't hear it well?
No.
I can comment it shortly.
Connie, if you can please stop the video. You need to stop sharing, and while sharing again, so sharing also the PC, the sound.
I can shortly maybe mention this at the same time.
Okay, okay, as you wish.
Okay, so what I will do, I will jump again to this slide, and I will just talk to you. So basically what I'm saying is that you can use any kind of tissue or sample that you're interested in, since it's autofluorescent measurements. Here in our example, we investigate Drosophila melanogaster and third instar larval stage, which you will see highlighted soon. So we will image salivary glands of this third larval stage.
Salivary glands have fascinated scientists for more than 100 years.
Salivary glands have fascinated scientists for a long time, with Walter Fleming drawing early images in the 1890s, focusing here on polychain chromosomes, which are specialized chromosome forms due to repetitive rounds of replication.
So these chromosomes allow high levels of gene expression in order to produce sufficient protein.
And I think I can jump to the next slide. So basically we are investigating this tissue, which is highly metabolically active. Sorry for this sound problems. I just go on. So I would shortly introduce how we prepare the sample. Before then we go over to the introduction of the system by Ali. So we use Drosophila larvae as mentioned. We dissect the salivary glands, which you see on the left, this little orange structure. Is it running now? Let's jump back here. No, sorry. Okay, here it comes. We're dissecting salivary glands from Drosophila third instar larval stage, and we expose them either to just PBS as a control sample, or alternatively, one of 2 metabolic inhibitors. The first one being 2-deoxy-D-glucose. And coming back to the scheme of glycolysis and oxidative phosphorylation from the very beginning, 2-deoxy-D-glucose, in short 2-DG, is an inhibitor of glycolysis as a competitive inhibitor of glucose, meaning that this should reduce the amount of ongoing glycolysis. And what we expect as a consequence is an increase in oxidative phosphorylation in cells and tissues. The second inhibitor is antimycin A, which you can see on the bottom. So this molecule blocks the electron, one of the complexes of the electron transport chain, which is complex 3. So if this blocks this sequential cascade, we expect a shift to more glycolysis in the tissues. We expose the tissues for 30 minutes to these different treatments, of course, individually and sequentially. And then after these 30 minutes, we immediately image the samples and perform subsequent data analysis. So just as a short sequence for the sample preparation, we investigate Drosophila. In the next image, you see a dissected tissue. It's very tiny, already covered with a cover slip. If the dissection has worked well, you can see a beautiful structure like this, which is a pair of salivary glands of Drosophila larvae. So the bit dimmer structure is the salivary glands, where you can guess individual cells also already. And these brighter adhesive structures are fatty tissue, which are also very interesting to investigate. So we seal the sample with nail polish in order to prevent evaporation during the measurement. And then the sample is ready for imaging. And with this, I hand over to Ali for the introduction of the instrument and live and recorded sessions of measurements.
Hey, Connie, thank you very much. And so just an overview, going through the instrumentation. This is a very simple drawing. The instrument we need is a normal microscope, which has stopwatch. Connie, you're on mute, I guess.
Sorry.
So for that, for the doing FLIM, we need a system which has a light source to excite a fluorophore and a detector which detects the fluorescence emitted from the fluorophore. And this is a normal microscope, and we need a PC to visualize the images of the microscope recording. Additionally, as I said, that's for FLIM, we need a stopwatch, we need a clock. What's happening here in the FLIM systems generally, when the light source is sending the light toward the sample, it's also send a signal to the computer and informs the computer that it starts to excite the fluorophore. And the computer starts the stopwatch. So, okay, go ahead, that's time 0. And then the detector detecting the fluorescence photons, the detector would send a signal to the computer and saying that, okay, I'm detecting 1. And the computer informs the stopwatch, stop. So the time between the excitation and the detection is the value that we are recording. And in the time domain FLIM imaging, FLIM microscopy, we use pulse lasers. So it means that every single pulse which is sent from the detector toward the fluorophore would actually also send a signal to the computer and say that, okay, I'm going to excite a molecule. And that one, if it hits a molecule, it would excite a molecule, depending on some other parameters, but we assume that it excites a molecule, and the molecule would be excited, and the emitting fluorescence, the fluorescence would be detected by the detector, and we do this acquisition several times, till the moment that we acquire enough number of photons to determine the fluorescence decay curve of this fluorophore. So what we see here, these dots that they are here, they are showing the number of the events. Number of the events means that's how many signals from the detectors came to the computer and say that, I detected something. And below that we have the time On the x-axis, we have time in nanosecond range. And as you see, it's like a decaying curve, and that's what we are looking for. It has also that in the beginning, a rising edge, and that comes from something that we heard a lot about that the last 3 days, instrument response function, which is the result of the imperfection of the shape of the laser pulse. That's the main contribution, and also that imperfection in the electronics, in the optics, and the cables that we are using, and the detector, and so on. And when we know the IRF, we measure the data, we can use multi-exponential decay functions to fit this decay that we have measured. When we do that, we get few values. One of them is the amplitude, and the other one is the lifetime. So let's assume that we have two components. So we have a short life component and a long life component, and each of them have specific contribution together to actually depict this decay curve for our fluorophore. And that's what we are trying to find out. For example, NADH, we know that it's been studied several times that it has a short lifetime and a long lifetime, and we need to find out what is the ratio between the amplitude of these two, and that's an indication for the metabolism states of the cells that we are going to study. So in this session that we are using Leica SP8 FALCON system, it's a complex system, as you see that here. So our system doesn't have the STED laser, but what we have, we can do a fully spectral confocal imaging, and for sure that confocal FLIM, but it's not really that's our interest today. So today that we would use two-photon excitation to excite the NADH and FAD inside the tissue, and for that we need the infrared laser, which would be guided toward the sample and then focus on the sample through the objective, and then we have the scanner unit to scan it. So that's a point scanning system, and the fluorescence on the back so would be collected by the objective and then sent to a NDD, non-descanned detection unit. So this unit is also the so-called 4-tune. That's the name of that. It's a fully spectral unit for the non-descanned detection modularity, and here that we can have also a spectral imaging. So this is how the system look like on our optical table. So the infrared laser would emit the light and then passing through this black box. It's black box, and then the laser is guided toward the scan head. It would be scanned and whatever is happening there, and then sent to the microscope stand, and where there are the objectives and so on. So we have a very short video just explaining so a little bit about the system, and just play it here.
Ali, same problem. We hear it, but we don't hear the sound of the video.
Okay, so I stopped sharing and I started again. I think that I have done it, but seems I wasn't. Okay, so screen sharing, share computer sound, and share again. There's two output line, one fixed line and 1045 nanometer with the average power of the system is equivalent with a multi-photon laser. It's actually infrared laser, but yeah, just turned out to be famous for multi-photon laser because we use it for multi-photon microscopy. This laser offers two output line, one fixed line and 1045 nanometer with the average power of larger than 3 watts. It's actually a strong laser, you see. And the other one is a tunable line, which you can tune it from 680 nanometer to 1300 nanometer. The average output at 900 nanometer is about 2 watts. This enables us to do simultaneous multi-channel multi-photon imaging. The emitted infrared laser travels through this black-red box, which actually, we just call it black box. We don't want to know what is inside that. Just it's enough to know that it's shaping the beam and it's modulating some factors of the laser line and so on.
but then everything would be just guided toward the scan head. The lasers are coupled inside the scan head, and then they are guided toward the microscope, which is focused via the objectives. So we have a set of objectives designed for specific applications. So we have two objectives now installed that they are dedicated for multi-photon imaging, and we have other objectives that they can be used for confocal microscopy. The excitation light would be splitted from the detection of fluorescence that's we are interested to collect it using the AOBS. The fluorescent light would then travel through the pinhole, which can be adjusted to have optimal confocal imaging. And then it would be guided toward the detection unit. So the internal detectors offer a fully spectral confocal imaging. So what we have here, there are 5 detectors. In addition to the internal detectors, this system is equipped with an external 4-tune detection module. 4 HyD detectors are installed in a non-descanned detection or NDD configuration, where the spectral detection ranges from 380 nanometer to 800 nanometer.
Okay. So I think that we had some questions in the chat. Gerhard Holz is asking, oh, okay, so it's already responded by Connie. The point here is that this is, Gerhard, this is the maximum output of the laser, and also it's not what we are using. So 1045 nanometer is not the laser beam we are using for the imaging. And okay, so no more questions so far. Okay, when we switch on the system, this is how you see that. So we have 2 screens. One of them is for the acquisition. The other one is we have the control on the FLIM module that we can do the analysis and also play around with some of the parameters in the acquisition. And just having a little bit the detailed look about that. This is how it looked like. You might have seen it since morning. That might not be new for you. What we have on the right side is a control on the microscope components. We can also design the experiments that we have. And also that in the middle, we have the control on the lasers, detectors. We can choose which detector we want to use. And also that's the detection range, as well as the other components which are in between. On the right side, we see the live window. So that's a multi-photon image. That's we see that while we are acquiring the images. On the second screen, anyhow, we see the FLIM module. So it's also divided into 3 sections. On the left side, we have all the controls and indicators that we need to decide which kind of analysis we would like to do and how we would like to do that. In the middle, we have the images. So that might be, you can change it with the fast FLIM. By default, that you see the fast FLIM. And also that we have these sliders to adjust the histogram, either the lookup table for the fast lifetime or the intensity. That would just change the brightness of the image that we are seeing. Below that, as you know, that we get, when we stop the measurement, we get the fast FLIM histogram. That's kind of distribution from the fast FLIM images. And on the right side that we see the decay curve. And if we do the fitting, which is on the right side, the right column, that we can get the fitting curve as well as the residual, which here we see that it's not really nice what's happening with this sample. We will see that. One thing which was coming up regarding in the last few days, it was a question about the fast FLIM. What is a fast FLIM? That depends on which system you are using, but there are 2 explanations that I know. One of them is that when you acquire the data, you have the arrival time of the photons. So when you average out, you have to find out the mean arrival time of the photons per pixel. You can estimate, you can estimate the kind of fast lifetime. It's not exactly the lifetime of your component, but it's kind of like mean arrival time of the photons that are coming. There's nothing calculated. Another way is that you have your photons and also that you know that how long it takes that they are reached to your detector. So you have all this information, temporal information, and you do a fast 3-component fitting to your data. I don't know really what is the detail of algorithm. Usually it's not discussed with the customers, but as far as I know, that's this is how it's done. So it would be a 3 components, decay fit, very fast one, just to visualize a fast FLIM image.
Ali?
Yes.
There's a question from the chat from
Yeah,
Pavel.
sure.
Is fast FLIM the same thing as rapid FLIM referred the day before?
No. Rapid FLIM is a different way that how you treat the information that you have, the temporal information that you have from the sample. But I guess that tomorrow that we have the session for the analysis using SinfoTime from PicoQuant, I think they can explain it better than me. That's what is really the difference. But as far as I know, rapid FLIM is a fast way to acquire FLIM images. It's not really about that's it's different than fast FLIM. So fast FLIM is like that's how to visualize the kind of temporal information very fast to give you a rough estimation what you're looking at in the lifetime domain. Gerhard is saying that I'm afraid there are various definitions of these terms depending whether you use time domain or yes. And regarding frequency domain, I don't have experience. I really don't know the answer. That how does that would be? But what I said, it was just limited to the time domain. You're right, Gerhard. So if we don't have any other question, then I just go through that's how we think that the proper FLIM experiment should be done. First of all, that's we need to know the system. So what parameters affect the FLIM data acquisition and analysis? It's not only that when you see the decay curve, then finding out what is the best or what is the ideal chi-square for your fitting, how your residual look like. It's also important to know what are the components on the way. So we need to know a little bit about the optics as well. I know that it might be a little bit boring, supposed to be application. That's why we have set of those experiments, but it is necessary to talk about this as well. And the other point is that we need to understand the analysis parameters with the standards for. So this is I'm going to do that live in a few minutes. So playing around with 2 dyes in solutions. I have just prepared stock solutions. One of them is Auto 390, which has a lifetime of about 5 nanoseconds. The other one is Auto 425, which has a lifetime of about 3.6 nanoseconds. So I would just later on go to the system and then trying to find out and understand about my system, how actually it is behaving. Another important point is that we need to measure the instrument response function of the system. When you are doing 2-photon FLIM, I like usually that's to use quench dyes like quenching erythrosine B with potassium iodide. That works for single photon FLIM as well, like confocal FLIM, or that you can prepare like a thin layer of the urea crystals on the cover slip and imaging, so the second harmonic generation from them and finding out what is the IRF, which typically that would be the shape of your laser pulse. And also the last step, which is very important to pay attention, is that plan milling full control experiments. So it is not really one-to-one when you have your sample or your fluorophore solution and in living sample. So we know that even the spectrum changes. The lifetime is even more sensitive in that term of the changes. But it is important to have understanding, if it's possible, that what is the lifetime of your fluorophores in solutions. The other one is that assess in vitro experiments if feasible. So for example, that for NADH, there is a publication by Melissa Scala and her team explaining doing in vitro experiments trying to measure the lifetime and the fraction of the different components, lifetime components of the NADH in solution when it's actually interacting with different enzymes like lactates, dehydrogenase, or malate dehydrogenase, MDH or LDH. So if it's feasible, please do that. So today that we have not done it. So in the experiment design that we did, and but I think that for these kind of experiments for the metabolic FLIM, it could be necessary to look how NADH lifetime, or FAD, even if it's possible, lifetime is changing in vitro. And do not think about any limitation on negative controls. So whenever you think it's needed, don't just ignore it. Do the negative control. Negative controls are very important. That's I might sound like that's it's super annoying and difficult to do this measurement, but we know in biology, in life sciences, negative control are very important. But in FLIM, there is another level of importance to that. So it's very important to play around with different controls. It's only not negative controls, so we need to have different controls with the for our experiment. So now I need to move out of the office. So I'm sitting in the office because in the microscope room is very loud. You will realize that when I go there. So just give me a few seconds. I walk through the microscope to the microscope room, and I see you there. And meanwhile, if there is any questions, so you can just write it in the chat, or Connie, that's I'll just get back to you with the answer of the questions.
So yes, I'm happy to take any questions if you can, if they come up, don't hesitate to ask.
So in the experiments with NADPH that you're usually doing, is it really important what the actual lifetime is, or is it mostly comparing before and after something, doing something?
I think, or in my experience, is that the systems actually differ a little bit in which lifetimes you calculate in the very end. So yes, it's rather a comparison of treated and non-treated samples in your experiment. So depending on how your system is set up and calibrated, I don't think one should compare absolute lifetimes. Yes.
Which leads me then to the next question. If it's just only relative measurements, do we need all the calibration stuff?
There is some discussion of this in the field. You can try to do, as Ali has mentioned, and there is labs who have published this. You could, for example, say if you assume that your NADH is composed of two lifetime, you can actually turn on my video. NADH is composed of two components in a relative proportion to each other, then the ideal controls were, for example, what's the one extreme, what's the other extreme. NADH alone can be measured easily in solution. However, you always have the in vitro situation in any kind of buffer, which you try to simulate your natural physiological conditions. And we've heard from a talk before also that, at least in my experience, you never reproduce the lifetime values that you obtain in a living biological sample cell tissue. But yes, the discussion is in the field, and you can also try to simulate, for example, if you know which protein your NADH interacts with, then of course you can simulate this in vitro also. But in a living cell, NADH interacts with numerous different proteins, enzymes, and so on. So you always see a mixture, a mean of these individual contributions of different protein interactions. So I guess you can always try to simulate this to a certain extent. I don't think you get to perfect values. I've seen some of the experts also on the field. If anyone else has any feedback on this, Angelika is around, I think. Amasi, Svetlana Kalinina is an expert on metabolic FLIM. So if anyone has a comment for the chat, or turn on your camera if you dare, feel free to answer this question. So yes, this is certainly an interesting aspect of these measurements.
Okay, Hela is saying that's why we prepare the salivary glands. Can we measure in vivo as well? The larva is not that transparent as far as we know that. So it's just a matter that's transparency and how we can image that in a light microscope. Otherwise, if the sample would be transparent, yes, we prefer to do that in vivo, but not with this sample.
There isn't
And
even, sorry?
And yes, Connie, go ahead.
I just wanted to comment on the in vivo, in vitro situation. There is initial publications on in vivo situations. The lab of Svetlana Kalinina, they investigated tumors in living mice. So they have kind of a way to assess the tumor, which is just underneath the skin as far as I remember. So it's somehow accessible, and it's kind of an in vivo situation, or it is an in vivo situation. For insect tissues, this is quite tricky, since insect tissues tend to be melanized, chitinized, and these are beautiful autofluorophores, which always give a strong background. But of course, the idea is a challenge to somehow go for this at some point, yes. But this was only a relatively simple, small test experiment here.
Okay, do we get the other questions? Paddy Day is asking, when you speak about controls, do you mean the usual negative and positive experimental control, or we need extra control related to FLIM experiment? So, Connie, would you want to take it? Go ahead with the answer to that. Connie? Can you hear me?
Sorry, no, I unmuted myself.
Yes.
It was just the point that we discussed earlier with Stefan. For example, one control measurement is, well, you basically can't have any living tissue you can deplete of NADH or FAD or anything like this. What could be controlled is actually something like the little test experiment that we run, for example, where you try to manipulate the metabolism, hopefully in a well predictable way. And then you measure what is the outcome, how do you change NADH lifetime patterns with this. And of course, you can have the in vitro situation, as more and more labs are doing and publishing this along with their data, where you try to simulate the interaction that you think you're investigating in vitro.
And Kao is asking, do you have to measure the IRF every time before a real experiment? The answer of this question is that when you know your system and how the acquisition and analysis is working, I hope that the background noise is not much, so our air conditioning starts to fly again. So it's working on the full speed now. And okay, the IRF measurements, as also that Stefanie Camp Peterson mentioned on the workshop, in their workshop, is that they do that every single time that they want to do measurement. I think that every day, so that you should do that. The point here is that it is necessary to do that because you want to do the analysis using the calculated IRF. So I would say that the answer to that every time before the experiment is that what the use of the IRF you have. So if you want to look at your IRF and then finding out how your system is working,
because
and if
the IRF will
okay, so if you want to assess that's actually the quality and about your system and the FLIM system, that's one thing. And if you want to use it for analysis of the data that you acquire, that's another thing. So we will see that in some point that's in our system that we might not need to do IRF measurement every single time that we do the measurement. I think that Mariano had some points about that.
sorry, excuse me, I missed up the question.
So we were just discussing here whether you do need it when you want to do a phasor analysis, and I think probably not.
Yeah, I'll say in my case in the morning, as I said, I'm not really interested in knowing the precise lifetime.
Okay,
but whatever it will do is just introduce a small bias, but the bias will represent everyone. So.
And yeah, so Amazi is also that spotting the IRF is measured when you change the objective lens. If there is no major change in the electronic or optical configuration, you can use IRF for at least a week for the same experimental conditions, excitation, wavelength, lens, etc. So there is another question from Delgadil, chemical question, cell fixation with PFA, for example, can change the lifetime of NADPH. We have talked about that. So NADPH is also that's it's a fluorescent molecule at the end, and changes in the microenvironment of the NADH, changing the lifetime of NADH. That's in principle. But there has been publication and there has been discussion that's actually they have measured and they found out that when you fix, for example, the tissue, they don't find really much of difference. But in principle, I think that's yes, there are changes, but that might be not really huge. I have not measured it so far, so I know that there is this discussion that in the community regarding that if it's really changing or not. And another point that there was a question that while I was moving, I just heard it that Stefan was asking if it's really necessary to really measure exactly the lifetime of NADH, the short or the long component. Depends how we would like to assess the data. If that's very important, the exact value for the lifetimes, then I would say that fixation changes. But if we are flexible in a range, so fixation, there are publications saying that it's not really affecting a lot the lifetime of the NADPH or NADPH. And
there's a comment from Amazi, which I think is exactly the point. I mean, for metabolic FLIM, you're measuring live specimens. So actually you want your sample to be as lively and happy and natural as possible. So this kind of contradicts a bit the idea of fixing the sample. But there is data also from the Amazi Periasami lab, right? From host Wallrabe, for example, testing certain fixation times and how that affects lifetimes and so on. Yes, it affects, but maybe it's not the best thing if you want to study a happy living sample.
Can I say something?
Yes, sure, go for it.
Yeah, so we did a lot of experiment on the fixed versus live cells, particularly for FRED. Because if you do RNA on GFP labeled proteins, RNA, it's difficult to use your live samples, and we had to use a fixed cells. We did some FRED work with RNA and the protein interactions. In that, we have to test whether really the fixed cells changes the lifetime. We found out that it changes the lifetime after a week. It again depends on the fixative, depends on the specimen. We used different proteins, different cell line. All those things tells us that after a week, it is an issue. The lifetime decreases. The fluorophore quenches, the fixative quenches the fluorophore molecule. But for NADH, not a good idea. If it is environmental changes. You have to use a live cells for metabolism measurements or NADH signal. We did fix the cell and tested that. Just, when you fix it, it maintains at the time of fixative. Then when you can see some lifetime value, but it is decreased. It's not 2.4 nanosecond, it is about 1.6 or 1.8. Then after the second time you excite, there is no NADH molecule. Just one excitation goes away in the fixed cells for NADH measurements.
Thanks, Amasi.
Thanks, Amasi. Okay, so I have shared my screen with you. I just checked that which one I have shared. Okay, great. What you see now here is the acquisition software. So it's been already discussed about that. I'm just going to talk about few things which are important for FLIM measurements. First of all, when you switch on the system, we need to turn on the lasers. So we are going to use MP2 laser, so multi-photon, the second beam of the laser, which is tunable. And there are a few things which are important. First of all, I'm just going to open the shutter for both of them, because one of the laser lines, so the fixed line, is used to as a source of the clock. So I can go a little bit in details, but if there would be any interest. But anyhow, if there are, I think that there is Martin from Leica also that's in this session. Martin, if you think that would be necessary, you can just jump in and explain regarding the 1045 and how this laser is actually going to be used as a clock for the FLIM measurement. But we need both of them. The other thing which is important is that to pay attention to the GDD curve. This is specific to the objectives, and that's a kind of correction because of the dispersion of the laser pulse in the optical path when we are doing the imaging. So we did the measurements and imaging with the 25x objective with the NA of 0.95, and that's a water immersion objective. So I choose the GDD curve that we have and close this one. So that's I'm ready. Now I need to choose which laser and which wavelength I would like to use. So just activate the laser and open the shutter, and with the mouse wheel, if I go here and just rotate the mouse wheel upward, I can increase the laser power. And when I have some laser power rather than 0, so we can see that's directed toward the objective, and then the specimen. Down here, below the objective, I have the control on the detectors. And I'm not going to use the internal detectors. I'm using the RLD, which is actually just a nickname for those 4 NDD detectors that I've just mentioned. And so here I see that we have 4 detectors. And fully spectral, I'm not going into details of that. It's been already discussed. And we can choose 1 or 2 of the detectors and doing the measurement. So I'm choosing the second one. And on the very first experiments, I'm going to have auto 425 in solution. And then I would like to do some measurements with that dye. But before that, let's talk a little bit more about how we are designing the experiments. When you have the FLIM module activated, you see the FLIM window. There are some possibilities here that how you can apply that to your FLIM measurements. And usually that I use the frame repetitions, and depending on the sample and also the acquisition and the brightness and what I would like to measure. So that can be something between 1 to whatever is required, but not really so long, because in some point it doesn't make sense. And also that down here, what is important is about the frame size and the speed. So the speed that here is that how many lines of pixel that we scan per second. So when we say that 400, that's the speed, that's 400 hertz, means that in 1 second, 400 lines of the of pixels would be scanned through the sample. And that leads to the frame rate of 1.3 seconds for 1 frame. As I said, that's also that in another workshop I mentioned that there is a discussion that it's a relation of these two. That's almost all the references that I'm mentioning that here, and also that Connie mentioning that we have prepared them on a folder, and we can share this information with you. So then link to this publication, and then you can read them through. So from different reviews, different works on the NADH, FAD, and also that's a different method to acquire them, and so on. So don't worry that we can share this information with you later as well. And then down here, so we said the SAC, we don't mind about that. Another thing is that we come to the fact that we would like to do NADH and FAD in 1 sample. So there is a publication, there is by Angelika Ruck and Wolfgang Becker, that so that's the Wolfgang that's talked about, Mr. Becker talked about that 2 days ago regarding simultaneous imaging of FAD and NADH in the sample using dual beam infrared laser. And that's one way. And the other way is that there is a suggestion and also publication from Mamazi lab that's saying that you can excite both of them with one laser line, and they take in different ranges, and then you have really negligible contribution or the bleed through of NADH to FAD. However, that's in our system, we cannot do simultaneous excitation because we are using a tunable laser, and it needs time to tune between two wavelengths. And I think that's not thinking. So I have done rough counting. It takes about 16 seconds for us to tune between 740 to 900 that later on we are going to use. Anyhow, so I'm going to share the other window and talking a little bit about the other part of the software, which is the FLIM software. Here what we have on the right side, we have different options for different analysis. If you have FRET, we can use a FRET module. We can do pattern fitting, which has been discussed also that before in the talks. We can do phaser, tau stat. We don't have stat laser, so tau stat, we don't do that. And we can do the ROI based, ROI based measurements analysis. But what's today, that's we are focusing on that, it's a FLIM. Down here, we have the region of interest. It's very interesting, one important part when we are doing the NADH analysis. And then we have another FLIM window in the FLIM module that would show us that how many repetition that the image is acquired, and if we would like to do the binning in the pixel or the lifetime binning and so on. Down here, we have the time gate. So actually we can adjust the range of the time that we would, decay time that we would like to do the fitting on the histogram here. And also that sometimes it might be necessary that we have sometimes a bump from the back reflection in our system, depending on the setting that we use. So if you have some kind of abnormality in your decay curve, there is a possibility you can add a time gate and actually discard that small part as well if needed. Typically, it's not really needed, specifically when we do the multi-photon FLIM. So I just delete this gate that I have added, and I've sent that to the full range, no changes. And also the intensity threshold, that changes the intensity, so we can set some threshold to remove the background to avoid any contribution of the background in the analysis, and also that it would just be reducing the number of the pixels that we do the analysis on them. Special setting, we don't use it, not talking about that. And it's also been well described. That's in last two workshops. Here, as I said, that's you can have the intensity image. When you do simply acquire the image, you see the fast FLIM, and then you can have here where you are at the intensity, you have the pixel intensity histogram, but it's actually showing the distribution of number of the photons per pulses that have been acquired across the field of view. So here, that's below on below the images on the right side, that we have the decay curve, that later on that when we do the fitting that we would have other information. And on the right side, we have the fitting column. So here, that's all the parameters and the controls that we need for fitting are sitting actually. There is one option here is saying that select fixed model. When I click on that, there are two options. N exponential reconvolution, that's the typical way we do the fitting. Yes, Mariano, so I can show you, it's not really that, interesting, but sure, I can show you that how the phasor plot would look like. So the N exponential reconvolution is that when you do the IRF and the whole data that we acquire, we measure data, the measured decay curve is actually a convolution of the instrument response function with the sum of the all decay curves that we have for every single lifetime component. The second one is called the tail fit. So I'm not going really in details on that, but what's happening with the tail fit is just say, okay, It's actually ignoring the rising edge, and also that's the beginning of the decay curve that we have measured. So what is the issue here? If you have very long lifetime, it is okay. You can use this tail fit analysis. But if you have short lifetimes, which are important for you, and we would like to measure that, then in this case, that's by just ignoring the beginning of the decay curve, we are actually throwing out the information about the short lifetime. And then this one would induce an artifact in the analysis we do. So that's why in our case, for the metabolic FLIM, we just focus on the reconvolution fitting. How do you mean with short lifetime? Any range? Okay. Carl, I don't mean that's why short lifetime. So in our case, we are talking about few hundred picosecond. So I'm not sure if also that's 1 nanosecond. I have not really measured with a dye with a kind of like around 1 nanosecond lifetime. That would just affect as well. But in principle, that's with the tail fit, we are focusing on the tail of the decay curve rather than the beginning. And that would just give statistically higher weight to the longer lifetime components than the shorter one. So we need to pay attention, and also that's be careful by using that one. So it really that's the answer of this question, is that how our system look like, how it is working, what is the IRF shape, and so on, as well as what I am measuring, what is the fluorophore. So you need also to know your sample. There is no specific answer, but short, in our case, I mean something between 250 to about 500 picoseconds. And Sandra is asking, what happens to the fitted lifetime when cutting out a regime within the decay histogram? Can we make a comparison? That's a very good question. I'm not sure really what happened. What I guess is that they interpolate this missed part that we are just going to delete it. So with respect to the other measured data that we have, what's exactly what is happening to that information, I really don't know. Maybe that's a person that's from the Leica, so they can maybe jump in, or somebody who has already experience with this multiple time gating. That would be interesting to talk about that. So that's how I use the time gate. I know that there are other applications for gating the time, but this is the use that we have for gating. So that I use it here, because also that there is this reflection filter which is doing the same thing. So it's actually know how the system is behaving, and it's applying so removals, because in some cases we have like a peak here, a secondary peak here. We're coming from a reflection, back reflection, as far as I know, and it's just deleting this part of the decay curve. So if necessary, I'm using the time get option for that reason. So what else do we have here? We can also, that's choosing how many components our fitting should have, and that depends, that's been already discussed several times, depends on the decay curve, and also that's the components and the fluorophores that we are studying. There are some values that I would like to talk about them. So I just go to 2 components. And first of all are the lifetimes. So the lifetimes we know. As I showed you in the presentation, we have this formula for the multi-exponential decay fit, and also that we have seen it here for the reconvolution, that the amplitude is a contribution or the fraction of different lifetime components. Tau 1 and tau 2 are these lifetime components, so they can, one of them can be short, long, they can be very close, not too close. That's actually comes when you are using more exponential components than necessary. You would get usually 2 lifetime that are so close to each other, and that's an indication that you are using so many components in your decay feed more than necessary. And just something which came up during the discussion was the question about the intensity and amplitude. So I can shortly talk about that. So just switch off the detectors, increasing the light. So I will be back. Okay, can somebody tell me that you can see the board that I have? Yes, okay, good. So since this question came up, I think that 2 or 3 times, I'm just going to briefly say what I think regarding that. So let us...
Maybe I just interrupt your screen sharing then, because then the image with your board will become bigger.
Ah, yes, sure, thank you.
I'll do it.
Okay, great, good. So I can just double click on my video as well to see that what I'm doing, and if you see that properly. So view, and that would be speaker view. Okay, good. So here we have our decay curve. So I just ignore the beginning part, which is actually rising up from the IRF. So I just draw that very simply. That's our decay curve. And when we do the analysis for the decay curve, we need to pay attention. What is the intensity? So that's the difference that we are doing this measurement. What is in the FLIM important for us is the lifetime components and the contribution in the decay curve. So if there are more than one, if there are several of them. But intensity mathematically is the area below this decay curve. So that's the intensity we are measuring. When we are doing normal fluorescence microscopy, what actually we are measuring is the intensity. It's the area below the decay curve. And when we have 2 components, for instance, a short one, which would decay like this, and a long one, which would decay like this. So what I understand regarding that's the difference between amplitudes for different components, short lifetime component and long-term component, it actually would resemble if I do the fitting. So that would be the decay curve for the short component, and that would resemble the decay curve for the long lifetime component. So the area below this curve would be the intensity which is associated with the short lifetime, and the green one, the area which is actually, it's quite difficult to show that, but yeah, this area which is under the curve that belongs to the long lifetime is resembling the intensity that which is associated to the long lifetime. So what is the amplitude anyhow? Amplitude is where that your decay is actually dropping to the 1 over the E value. So that's where that your lifetime, and also that over the time the amplitude is dropping down as 1 over E. So I just move this one back. Okay, I hope that was clarifying something regarding the amplitude and the intensity. So the intensity is the area below the curve, and the amplitude is the value that we have when we are going to define the lifetime. That's the amplitude, the maximum in the decay curve. So I'm just going to start my screen again here. So here is that we have the maximum, the count rates on our decay, and then when it's dropping down to 1 to this value over E. So this is that we say that, for example, a lifetime is 3 nanosecond, and then the amplitude would be something like here in this case, for instance, like 100,000. Okay, so let's continue with that. Then we have two values, mean lifetimes. One of them that we are interested in, that is the amplitude, it's amplitude weighted one, which is the mean lifetime, which actually weighted by the amplitude. Is like that's amplitude 1 multiplied by lifetime 1 plus amplitude 2 multiplied by the lifetime 2 divided by the sum or total of the amplitude. So when we do the fitting, we get all these values. So we get the sum of amplitudes, we get amplitude 1 and amplitude 2, and one actually can calculate, even if you would like just to check it yourself. You're interested in that, you can calculate this mean lifetime amplitude of data. Okay, after all of this talk, that I would like now to put some of our solutions. So it's auto 425, and I would like to see the behavior of our system. So I would like to show you something, because this one also that came up. I hope that you hear me well. I would like also to show you something else, because this one came up as well. So I stop sharing that you would see my video well. The question was that when you are measuring the IRF, if you're just putting a droplet of the liquid on your cover slip, it would be evaporized or not. And Steffi was explaining that it's the measurement is so fast that you are not going to have any problem with the evaporation. But since I might be, it might take a little bit longer for what I would like to do, what we are doing in our facility, we are using, I'm not sure that it's already feasible, so not really focusing properly. So we are using the PCR epi tips, the door of them. So just cut it out, and we are using that one as a container. So you can add like 50 microliter of any solution inside them. And just put this one upside down on the cover sleeve, and then placing the cover sleeve on this microscope. So then go back to this page. Okay, sample is placed, and what I'm going to do now, just changing the wavelength to, let me share the first screen now. Actually, for making that easier from now on, what I would like to do now, you're seeing my first screen, I'm going to minimize the acquisition system, acquisition software, and just avoiding the live view. So it just takes a few seconds to do so. Because I'm not interested to see the live view, I can see the live view also that in the FLIM window here. And then bringing the FLIM window also here. Okay. And a little bit of moving this one out, and also video. So now I might be able to see the chat as well easily. Yes. So they're coming here. Okay, so minimizing this one to having everything in one view. Is that okay?
Yes, looks good.
Okay, great. So I hope that everyone can read the details. The numbers are not really necessary, so I can talk about them. If they are necessary, I will explain them. Otherwise, it's not really something very, very special there. So here, that's what I have. It's now the laser is set at 740. I'm going to change it to 900 nanometer. This is what we are going to use for FAD, for instance. Or I can simply type in 850, and it takes few seconds for the laser to tune. So this is up to you. As you see that simply you just type in, or simply you can drag it. I would just stick to 850 for this measurement, and also that using the second detector. And as soon as that it would be tuned, that there is a laser line, and I'm going to acquire a signal in this range. So it's from 470 to 560. I just set it by like that. So it's not really nothing very special. So it's not specific numbers that I'm putting. But since I have only that's this Alexa 425 solution, so that's a good range. So I can just start with some live images, see that's what we get out of it. So when we are, oh, cool. So already that we see the decay curve, and what is important here, that's this like noise, like salt and pepper noise that you see, is actually our signal on 512 by 512 pixels at the rate of 400 lines per second. What is very important is also that this pixel intensity histogram, I can increase the laser power to increase the number of photons per pulse. But the question always is that necessary or not? So do I really need to do that or not? Another thing that's here that we see on the, when we are on the live mode in the acquisition software, what we see here, it's frame by frame. So that's actually what the decay curve that you see here is that for one frame. And here what we see is the number of the photons that we are acquiring per frame. What we see here is not for one pixel. That's what, because some of the softwares that the acquisition FLIM software, they show you the decay curve of a pixel, or after the binning on the lifetime, or the pixel binning. But here what we see is the whole field of view. So that's very important. And when we are on the live mode, it's that like frame by frame at the moment that the field of view that we're seeing. I'm quite okay with something like ranging between 2 to 5-6%. And this value is saying that I just stop it. So the MP laser repetition rate is about 18 megahertz. So it sends 18 million pulses per second to the sample. And this value that we see, that the photons per laser pulse, saying that how many photons per pulse that you are going. So Falcon is a fast system. It's actually suggesting that we can acquire even sometimes not recommended, but more than 1 photons per pixel. So what I have as a sample is 10 micromolar auto 425 solution.
So there has been a question in the chat. I think that's Louise asking, what is an acceptable range of photon counting? So I guess you've answered this question with this already. If there's still part of the question, maybe you can answer, or yeah.
I can play around with that, but what is the case here is that let me increase the laser power. So now I'm really in a very minimum. So I increase it, and then you see that there is a limit. So there is a red line that's showing itself. So I'm going to reduce also that the okay, that what was the sound? So what you see, it seems that I'm saturating the detector. So detector is not happy really with this much. So I can increase a little bit more. And in some points, yes, it is saturating. So the point here is that I'm not saturating the electronics to do the FLIM measurement. But what I see here, this line that I see, that's my hybrid detector. So the HyD detector, which is super sensitive, is not happy with this high intensity. So I would just decrease it. But as you see, that's the decay curve looks really less noisy, less fluctuating, especially that's here with the longer, when we go that in the longer time in the decay curve. But what you see that here as well, that's the characteristic of a decay curve when your lifetime that you are measuring is not perfectly matching with the repetition rate of your laser. So what I'm using now here is an MP laser, as I said, that is 18 megahertz repetition rate, which means that actually there is 12.5 nanosecond time between each 2 pulses. So that's meaning that we have 12.5 nanosecond to acquire the decay curve. That's the time that we have between 2 pulses to calculate, to measure the decay curve. What is the lifetime of the dye that I'm using? So nominally, it's 3.6 nanosecond. 12.5 nanosecond time between 2 pulses is not enough to completely record the whole decay of this dye. That's why that we see the end of the decay curve actually showing up in the beginning of the decay. It's like that's it's sending it to the next pulse. But anyhow, so we have algorithm that they can fit this residual from not that's incomplete decay curve that we are measuring. Anyhow, so what I can do, I was not able to show you what is really the limit of that. This stops at 1. So there is a red, it doesn't stop. It's actually, there is a red line at 1, and it's telling you maximum 1 photon per pulse. So meaning that you should consider something about 80 mega counts on your detector. And sometimes it's not possible when you have super bright sample. So like we had, we can use 2 detectors and then combine the counting from 2 detectors to 1. And in that case, that we can overcome of this limitation by overloading the detector. So the sensitivity of the detector is high, so it's getting complaining that it's too bright. By the way, I really don't need this high laser power. So where I was with 0.1, I was happy with that. That's not really much different. It wasn't really much different between that's what's at the end that we are going to acquire NADH or FAD. So I can maybe that increase it something like 7-10% on bleaching, but I don't think so. Or maybe it seems actually the signal drops down when I was at the high, but in the solution I didn't expect to see that super fast. Okay, anyhow, now I would like to do some measurements, actual measurements, FLIM measurement, and I would go for something like the frame duration of a minute, or I can use frame repetition of 20. So we see that 20 comes from the fact that we are doing our metabolic FLIM imaging with 20 repetitions. So I'm just going to start this acquisition. And then what is the next step is that playing around with the fitting parameters of the software. So talking about some of the parameters and what is important, what do we have, and how we can assess the analysis later on. This is a part of knowing the sample, knowing the system. So it's quite nice counts in the whole. We have more than now, it's not done, the measurement is not done yet. We have more than 4 million counts in the decay curve. And again, I'm saying that it's in the whole field of view. That's how this decay curve is shown here. And also that's what we see here is number of the photons per pulse. That's maximum is 10%. So it's every 10 pulse that we acquire 1 photon. I just moved to the fast FLIM image, and I'm expecting that's the fast FLIM histogram, which is showing the lifetime distribution across the how often that this lifetime value in the whole field of view is actually occurred. This is the histogram, the lifetime of the fast FLIM histogram, which is represented. And it's saying that there is a peak about something like three something nanoseconds. As we talk about that, fast FLIM is like a very fast estimation. And yeah, so now we are starting with the n exponential reconvolution and the exponential component of 1. This is what we are expecting to would give us actually the best result. I just click on fit, and this value that we are getting is about 3.5. And we were expecting something about 3.6. There is something here that's it's very important. So this adjustment on the peak of the decay curve. That's it's really a little bit mismatch that we see here. The chi-square is about 12. It doesn't seem really that work very nice. So what might be the reason? If I just go to 2 components, clearly it is better, and I'm getting a short lifetime. At the top of that, the value which I'm expecting to see that for my sample. So should I accept this one? And also the chi-square is actually, it's really nice. It's about 1. That's very good one. But I have this extremely short lifetime, which is, I'm not happy with that. So that might be because of some imperfections. So that's coming from the system. I don't know. But I was expecting to see something like this value in the very first hand. Is there anybody that would think that I should go for 2 component fit now? So you can just unmute your mic and then talk if you think that I should go for 2 component fit now. Okay, nobody thinks so. So let's see that I just go back to 1 component. I do the fitting, and here, so we have 2 parameters, IRF background and IRF shift. Seems that the problem here is at the top. So maybe that there is a shift between the peak of the lifetime and also the infrared, the instrument response function. But actually, it's not really solving my problem. And the IRF background, I'm not going to change it. I keep it to 0, because what is important here is actually when I let the IRF background to be fitted, it's going to eliminate the contribution of incomplete decay in the beginning of my decay curve. And I'm going to lose some information. So what was the problem? The point here is that there is actually not much of a problem. It's simply that how our system is behaving, and we see that in the rest of the analysis and acquisition that we are going to acquire, and see that one more thing is that our experiment, we have to pay attention to that. For example, that I did very similar experiment with Auto 390, and what we see here is a nice, very nice fit. It might be that actually my stock solution is getting old. I don't know. So I have not prepared today morning. So it's been there for a few days. That might be the reason I'm not getting really the lifetime and also the decay curve I'm expecting. So this is from Auto 390. So what I was expecting was about 5 nanoseconds. But on our system that we get sometime, almost always, we get a little bit longer lifetime. There is a discussion, so I really don't know the answer of that, but always that we get a little bit longer lifetime than expected. So this is what we are expecting from our system. So this one, what I did right now, is shorter lifetime, that 3.6. But still that's in the range of 80 picoseconds, and also that's really in the range of the resolution, temporal resolution of our system. So can be accepted 3.52 for 3.6 nanosecond lifetime that we are expecting. But as I told you, that's we know our system, and we are expecting usually longer lifetimes for the measurement that we do. So this is from Auto 390. That's a fresh stock solution that I have prepared a few days ago and did the measurement right away. And you see that the chi-square is about 1.2. And the lifetime is slightly longer than expected, but this is what we expect from our system. And also the residual looks very nice. And we don't see very specific residual. I also can adjust the range of the fitting. So to in the very beginning that we have this fluctuation, which I'm not sure that's where it comes from, but you can ignore the very beginning. Yes.
Hey, I have a question. I think it's easier.
Yes, sure.
So just by curiosity, have you tried to compare for the same solution and let's say the same laser or a different laser, comparing the dive detectors and the internal detectors, if you obtain the same results?
I didn't get your question, sorry.
Have you tried to compare the results, because this one you get it with the MP and the dive detectors, not the internal ones?
Yes.
I know you're the dive, yeah. Have you tried to also measure with the internal detectors to see if you get the same result?
Yes, exactly. That I was really thinking, frustrated. Why always I get longer lifetime than expected? It's like something like about ranging between 30, 40 picosecond to 110, 120 picosecond. Usually it's longer. So what I did, I used internal detectors. I got the same result. And I did single photon excitation, and I got almost the same result as well. So that seems this is a value that I get.
Okay, and you also try with because with the white light laser you can change the repetition rates. So even with
the repetition rate. Yeah, I went for Auto 390. I don't have the data here now, unfortunately, but I did with 20 megahertz. Because with the white light laser that we have a pulse picker, we can change the repetition rate of the laser. I did even with 20 megahertz to see the complete decay curve, since it's 5 nanoseconds.
Okay, thank you.
Yeah, sure. Thank you. Yeah, so this is the value that we get. And one thing which is also that I would like to try, that now I have this long decay curve, and I see that the measurement was not really responding well, and there is a problem in the beginning, in the very short lifetimes. What if I use the tail fits? So now I have long lifetime, so 3.6 nanoseconds is relatively long. If I use the tail fit module for fitting that, okay, tail fit, okay, and I do the fitting, the result is actually is as I was expecting. So 3.64. So this is what I have measured several times. And what I get here now is there is some contribution either of the IRF of the system here today, because today specifically our air conditioning is super active. And usually that's when it's super loud, it's because that's trying to compensate the humidity inside the room with relation to the outside the room. So it's trying to keep the humidity fixed, stable. And I think that we have this humidity issue as well. So that's also that effect that maybe, I'm not really sure that's why I couldn't get the proper value. But as soon as that I use the tail fit, so the contribution of the short lifetimes is just cancel out, ignored, and we are fitting to the decay curve toward the tail fits, to the longer lifetime, to the longer lifetime components. So in that case, that the value that I'm getting is actually more correct. But it's not usually the case. So I'm surprised seeing that today, that with the normal exponential reconvolution, I'm not getting the value that I'm expecting. Okay, so that's so far. I wanted to talk about that relating the analysis and also the difference between the intensity and the amplitude that came off these two. When we do the fitting, also that it is possible to use the region of interest, especially when I do with the solution. I usually like to focus on the middle of the field of view, not really acquiring the edges. As you see that here, it seems that there is some intensity dropping down. So we have one column of the pixels, which they seem like 0. So I just usually let's stick to the middle and defining a region of interest, and then I do the fitting on this region of interest. It's not always different value, but what we see here is that the decay curve is cleaner, so it's the chi-square is reduced, and that's why usually that when I do the imaging, I try to stay in the middle of the field of view. In principle, that we have a higher chance for optical aberrations as well when we go to the side of the field of view when we are imaging. So when we are using the optics, and also that's not really that using the zoom later on that we see that how does that work. Is there any question? Okay, if you have no question, that would be the time for me to go back to the presentation. Is everything clear? Okay, good. So I won't be at the system in the rest of the workshop. Oh, okay, there is one. Is there any advice to know if we need 1, 2, or 3 components? Yes. First of all, I know this, I know my sample. So I know that it should be 1 component. If it's not 1 component, either I have contamination or my sample is also what mainly I expect from the measurement I did today, because I prepared the stock solution that's days ago. And so there might be something wrong with the solution that I use. That's why I see the kind of weird behavior in the short lifetime. Also, there might be the chance that some components in change. So in our system that we use auto select option for different components that they can be on the optical path. Everything is automatically selected. And usually what I do, I check that this component, that this is a short pass filter, And also the filter cubes in the revolver, in the microscope body, and also that's the fluorescence turret, that what are the components when I do the measurement to make sure, correct? We are using the auto select for them that matches the best for the measurement we want to do, but we want to make sure that it's not changing from experiment to experiment. So usually that I, in the metadata, I check and make sure that the auto select is actually selecting exactly what we want. And also that's from 1 to 3, the chi-square. I think that it's also I just finished that from 1 to 3. So then you go, if you don't know your sample, you don't know that how many components that have, you go 1 and then you see terrible residual. Okay, let me show you. So I was showing one data, and here is the decay curve, and then I fit it with one component, and you see the residual is, it's actually, there is a systematic pattern there in the residual from your feet, and you can see the, okay, that's the same feet. So I just go to the reconvolution. You can see the systematic pattern in your residual, and specifically that here, this part. Moreover, that what you see is that your decay curve is not matching your measured data. So that's why, that's the point I go to the second component and then try to find out if it's a double exponential decay. Then I do the fit. And here, see that actually there is like a pattern, like a systematic pattern. The residual is not great. So now the question is that, do I have a third component, or there is something in my sample which is actually contributing to the signal, but it is not what I'm interested for? In my case, I can go to the third component and try to do the fitting, and I get very nice fit, but the values that I'm getting, they are not the ones that's expected for NADH, and this is actually NADH image. So I have the prior knowledge, and I do the measurement. I see that, oh, okay, oh, third component is necessary, but I was expecting 2. What might be the reason? The reason is that we are also considering the nuclei, which has non-metabolically active NADH in it, and that has a different lifetime. And we don't know, I don't know what is that lifetime, how actually it is, in which direction it's contributing. But for actual analysis, I need to discard the nuclei in my field of view and then doing the analysis outside the nuclei, only in the cytosol. So that's one thing. Chi-square is one. The second one is that you look at the residual, if there is any evidence deviation in the residual. So here, see that there is like a fluctuation between, if I just again start from 0.2. So it's like changing from -15 to 15 counts the residual. If I just zoom in here, that seems like there is a systematic pattern. So I see that here is a tiny deviation from the measured data, where I have in the whole field of view something like 25,000 counts, and we are talking about 12 deviation. That's true. It is said that we have to search for the chi-square, which is about 1, but not limited to that. So it's not the only parameters. So Powell is asking, the chi-square is not that good even with a clean sample. What is the general advice? How to deal with the bad chi-square values, and what do they say? So I was talking about that. The chi-square is not the only parameter. So I'm not saying that fit your data with any chi-square. So if the chi-square is 100 or something, that's a lot. But 18 as well, it's not really usually that's what we see here. And that's because that I have not discarded the nuclei. So usually that I get something between 1 to 4 or 5. So for the chi-square. That seems in contradiction with many advice regarding the chi-square. But what is important is that in some of the softwares that what is shown here is a reduced chi-square. That's a different definition than the chi-square itself. So what I have learned and I've been told regarding the chi-square in this software, the value is about 5, 6, even to 8 is actually okay. And there is a suggested minimum number of photos per pixel. It says suggested minimum number of photos per pixel to avoid overfitting or deviation. Here it's Dovidas is asking this question. Yes, in principle, you need to have good statistics to get enough data points in your measured data to calculate the decay using fitting algorithm. But in our case, that's what we are doing here. This decay curve that we are seeing here is a decay curve from the whole field of view. It's not from a single pixel. So I have not done pixel-wise fitting yet. So this is what I have to emphasize again. What I'm doing here, I'm just looking at the whole image to find out what is a good strategy for the fitting for later on. So do I stay to the whole field of view? Do I go for a specific region of interest? Do I go for pixel by pixel analysis? Do I stay to the mean of the region of interest and so on? So far, I'm just talking about that. What is the proper strategy for the fitting? And yeah, so when it goes to the pixels, yes, there are suggestions saying that there are also that simulation and measurements saying that you need about 100 photons if you have the mono-exponential decay curve to fit. If you have two, that goes higher about 2,000 or more. So in the range of few thousand, 1,000 to 10,000, there are recommendations. On another system that we had, I was using 2,000 counts. That was for the brightest pixel. But in principle, something about 1,000 to 10,000 is recommended. And then when it goes to the third component, the number is just exploded because the degrees of freedom we have in fitting with three components decay, it's a lot that requires so many data points statistically to avoid artifacts or miscalculation and so on. Would the pulse shape of the laser have an impact on the measurement in case it changes over time? Gerhard, that's a very, very important question. And I hope that the shape of the pulse, I hope it's not changing over time. So I always switch on the system hours before I do the measurements to make sure that the infrared laser is stable when I do the measurement. And I hope it's not varying. So at least the shape of the pulse is not varying over time. So Mariano is actually responding to that as well. And we will choose. Okay, good. Perfect. If there is no question missed here, I'll just stop my video and also screen sharing here and see you in a second in the office.
Maybe in the meantime, I can shortly comment on the number of photons collected from my experience. So I never had the impression that I can over collect photons. So the more photons you can actually collect, the better and the cleaner your results get. I mean, you have to keep in mind, at least for these metabolic measurements, you're looking at autofluorescence, you're looking at autofluor 4. It is likely that actually it occurs in not only these two distinct states, but maybe even the complexity is higher. So I guess the more photons you collect, the cleaner your fit gets. And I can just repeat Mariano's comment from this morning also. I don't think there's a fixed number which you can set as a limit, but the closer two lifetimes you are trying to disentangle, the more photons you need to actually properly do this. So this is something you also have to keep in mind, which I think is important. So it's rather a testing and checking certain proposed values and then see whether you can properly fit your data, whether the values you get make sense overall. Maybe also collect more photons and see if that improves also your fitting, or if that doesn't do much more, then maybe you're in a good range.
Okay, so here we are with the continuation of slides. Good. Peace and
Ali, can I make a comment?
yes, sure.
In the lifetime determination, quantitation of the lifetime signal, we do not depend on one value like chi-squared, one value like any information. So mainly you have to justify when you use the lifetime imaging systems based on your biology, based on your experimental condition, and that's how you have to determine. We cannot go with one number, say lifetime value. Lifetime value varies. You measure in your lab for the same fluorophore. If I measure in my lab with instrument, it will be different. For example, they measure lifetime in different labs. 3 nanoseconds cerulean, we measure always 2.8 nanoseconds. In lifetime measurement, the reproducibility of the results, reproducibility of the data is very important. You cannot compare with one lab to another lab. It may not vary that much difference between the lifetime value. And also you should always use some kind of standard to test the lifetime system, make sure you can reproduce the value. These are the very important points we have to think whenever we work with lifetime imaging systems.
Thank you, Amazi. Yes,
Yeah.
That's exactly the point. We are all measuring the data with one system, and that's one of the, I would call, limitations that we have on the FLIM microscopy, that we are limited to the correlative analysis and experiment. So we need to find out what is the condition that we have with our system and in our lab with our actually experimental design, and then keep it consistent through the experiments that we do. There might be slightly differences between lab to lab, instrument to instrument, and these differences can come because of the quality of the optics. It can be because of the different laser models or the companies that they are using, or different specification that any component optically has that's on the optical path. Plus, there is a software in between which is interpreting the data to us. So that software uses algorithms, and these algorithms, they are implemented from instrument to instruments, from manufacturer to manufacturers. So there might be the statistics and the mathematics behind that, they might be the same, but how it is implemented, and if there would be any specific filters applied or thresholding applied, or even the number of the iterations, the maximum number of iterations, that because when we are doing the fitting, we are doing the iterative, we are running an iterative algorithm. If the maximum number of iteration in one software is 10 and in another one is 20, we get different chi-square value. We might get a little bit different values also, that for the lifetime and the fractions of different lifetime components. So these are, we have to pay attention. There are reasons that's not specifically, they are related to how we conduct our measurements, how we do what we do. There are some parameters that's, as a user, we are not aware of that. So we just need to know our system at the best possible to know how to implement our experiments optimally. Okay. Amazi, I would be happy anytime that you think it's necessary and I'm missing a point, just come in and correct me or adding the points. I would be really happy.
Yes. Yes.
So seems that we have in chat something. Is that any different from measuring intensities?
That's for sure.
Yes, and but
Well, you've described this or the problems associated with the measurement of fluorescence lifetimes. But I think there are similar problems in comparing results when you quantify or measure intensities.
yes, it's just a matter is that when we are measuring the intensities, we get some of the information. So what we have is a gray value, and if we know how the number of the photons or actual optical signal is translated to the gray value in the histogram, then we can calculate it back. But with the lifetime data, it's not always clear, or at least I can say that this information is not always shared with the user, unless that you build your own system and you know what it is, or you write your own software. And when it comes to the intensities, I would say that it's a little bit easier because no fitting, no iterative algorithm to export the information is actually involved.
But you don't know what the fixing is doing in your detector with your camera.
Ah, that's a good point. That's very important point. Again, that's we try to limit the number of the variables in the experiment. But maybe that I can take this opportunity after your point and correct myself. The number of the variables that we need to make sure that they are not changing from experiment to experiment in our lab in lifetime-based measurements are more than the number of the parameters we have to take care in the intensity-based. Thank you very much for bringing that up. Okay. So I played around with some solution, and I hope that you're not bored. Actually, I had the feeling that I had a kind of like a competition between the air conditioning and me, who is louder when I was in the lab. And so my voice is a little bit changed. I hope that it's not this.
You won.
Yeah, but I'm not sure about the price. So I see that tomorrow. Okay, good. And so what I have done, extra, so not today, it's not going to be live, is that we have measured the instrument response function of our system, and also that's measured the lifetime of NADH in solution in phosphate buffer. And then I have recorded the videos. I'm going to show you the videos and talking on them. And if you have any question, I would be happy to address them. So let's go to the next. That's done. And here is a video that's when I have measured NADH. It actually doesn't have NADPH in that solution. It's only NADH. And let's see. Okay, it's supposed to maximize when I play it. Sorry, give me a second, please, because it's supposed to maximize when it's played back. The next one, no, none of them. Okay. Let's go to this again. Oh, yeah. That's it. So what is happening here? I'm going to set the laser wavelength to 740. That's the wavelength we use to image NADH. And using the NDD detector number 1 in the range of 435 to 485. That's the range, detection range that we use to image NADH. That's exactly the same configuration on the system that you use in the actual measurement in the tissues. So here, that's when I just adjust it on 435 to 485. So I just start live and gradually increasing the laser power till the moment that I get some signal. And clearly you can see, since it's only NADH in the free form, it has extremely short lifetime. So here is the decay curve, and it's decaying super fast, indicating a very, very short lifetime for the NADH, free NADH. So after that, it's setting that's up, just do the actual measurements for 20 repetition here. And also that's, so looking at the number of the counts that I have in the whole field of view, because later on I would like to see when I fit it, what is the value that I get. Okay, that seems quite fine. There are more than 200,000 counts, and we are expecting that NADH, when it's free, it would have only one lifetime component, which is a short lifetime component. And then okay. Then after this measurement, what we have done, we started to the actual measurement with salivary gland. So there is one sample. So what I have here as a sample is a freshly dissected salivary gland from Drosophila larvae on the 3rd instar stage. As Connie explained previously, just reminding you what we have here. And I'm going to image NADH in that one. So we're starting with the setting of the laser at 714 nanometer. That's a wavelength we are going to use. Opening the shutter, so you see that it gets this signing that you are opening the shutter or closing them. So I just double click on the wavelength number and then adjust that to 714. Then double clicking on the detection range, typing in the range that we are using for imaging NADH. So 435 to 485. Actually, what should I say, that when you look at the publication, there are different ranges that they are used by different groups, different publication. I think that it's not only that's how the system is actually, it was designed. It's also that they talk about different reasons that this is the range, which actually they have a clean signal and no contribution from other components and so on. So what I have here, I'm just, I would like to just stop it here. What I see here when I start to get the first signals, the first grasp of how the sample look like, I can see, first of all, this is my sample. It's super noisy, and there are a lot of pixels without information. And I am going to spend a lot of time to acquire these pixels that contains nothing. So that's why I need to change a little bit the strategy. What I do, I would just go for one round of imaging to find out that's how it is. So that's 5% laser power is what we use for actual imaging. So the last measurement we did in the lab for the laser power, it's at the maximum output of the laser power just on the back side of the objective, before the objective, was about 380 milliwatts. Sounds a little bit low. We need to repeat it, but we are using 5% of that maximum output. So this is what I did. There is a sample, so I need to locate the proper field of view. I need to decide that what is the proper field of view, what I'm going to see. So the ROI, that is important. And also that's what is the resolution I need, what is the speed. So that's the compromise I told you. That's usually in microscopy we have speed, then we have the field of view, and that one is actually correlated with the resolution you need, and also how long it takes. So, and also if you want to do 2D, 3D, and so on. Okay, just fast forward it a little bit. So this is the part that we don't want. So these are, seems a little bit with longer lifetime. They are the lipid tissues that we have, the lipid cells that we have. And here, this one, I had no idea what it is. That's why I reduce the laser power again down to 1% to locate the area that I would like to measure it. But how I'm doing that, here, if you look at the zoom factor, I'm using the zoom factor. So I'm just zooming in the field of view, in the center of the field of view. So if I zoom in by the factor of 3, I would get the pixel size of about 300 nanometer. Clearly, it's below the resolution that we can get with the multi-photon system. It's not optimal resolution, but that's not what really we are interested in. So not always necessary that I go for the best resolution, because it really depends on the biological question that I have. Do I need the best resolution that the system can offer? In our case, no, we don't need it. So what we would like to do is that to look at the metabolism state, but looking at the cytosol, we are not really looking at single mitochondria. So that's why I think that 300 nanometer pixel size is a good compromise that we can do. To have the proper size for field of view, to have 2 or 3 cells in a field of view, as well as to be not really with a terrible resolution as well. So the images have enough spatial information, as well as that we would be fast enough to get enough temporal resolution. So as I said, that we go for the frame repetition of 20. So we found this one a good setting for the sample that we are measuring for our tiny showcase experiments that we have designed for this workshop. But this is up to us. So to go for the frame duration or the frame repetition. So we go for 20. It's acquiring 20 images. What's happening here in the lifetime, in the FLIM module, it's accumulating all this information at the top of each other. But if we see that here I have minimized the live image, it's actually averaging in the multi-photon image. In the intensity image that we don't see that here, it's averaging out, but in the lifetime image that we are seeing, it's accumulating everything at the top of each other. So now we can see the clean decay curve in the whole field of view that has been acquired. And then we need to check this one for the analysis, for the 1 or 2 component analysis. That's what we have been, I've been showing a little bit just before coming back to the office. And then we need to check for the sequential imaging. So what is important here, so far I was talking about the NADH only. The second part is FAD. FAD was not our main focus. We just did it as a bonus. It's a very tricky experiment to do. But since that we have to switch between wavelengths using a tunable laser that takes something like 16 to 20 seconds to tune from this one to the other one, so we need to switch the sequences for the NADH and FAD between frames or between the stacks. So we cannot switch between lines, we cannot be fast, we cannot be virtually simultaneous in this imaging. But with the data that we have acquired, we realize that we don't have much of misplacement of the sample in the field of view when we are doing the measurement. So I think that there is a question. If you have a pulse picker and can change the repetition rate, should you go for the fastest pulse rates that will contain the entire decay curve? Are there other factors that affects your choice of repetition rate? Jessica, the point here is that the pulse picker, we have it for the white light laser. I am not aware of a reliable pulse picker or a commercially available pulse picker for infrared lasers. So that pulse picker that we have is for the white light laser, not for the infrared laser. On the infrared laser, it is running only on 80 megahertz. So we cannot change the repetition rate of that laser. So that's why that's the only choice we have. So we are limited to this factor. And another one.
However, I would like to mention, yes, this was a good point of thinking of a longer decay window. And that's what I've thought of. Fabian yesterday mentioned that the time frame timing between pulses should extend 10 times the lifetime you're planning to obtain. So in this context, yes, a bit slower repetition would probably lead to better lifetime data.
Yes, Connie, Hela is also mentioning there is like a feedback loop in your voice. But also that Mariano is mentioning that the point here is that you need to have the time between two pulses, as I mentioned also earlier. You need to have the time between your two of your pulses long enough. So Fabian, Connie is saying that Fabian, you almost mentioned that's 10 times longer, but I think that's something like 5 times longer. It's quite enough to see the complete decay. And Mariano as well is responding to that. So other than a lifetime of your dye, the acquisition speed will definitely determine the choice of repetition rate. Yes. Perfect, Mariano, for mentioning that. That's another parameter that we need to pay attention for that. There is something which is very important, is that how long the laser standing at one point to acquire the photons, that you would have time to acquire the photons for 1 pixels in your image. That's called pixel dwell time. And one thing that actually it was very important to talk about, that's there are different recommendations. There is a publication, there is an application note. I mentioned it, we can share it with you as well. And this application notes does the calculation and simulation and suggesting that, for example, that now I'm doing 400 hertz imaging with 512 by 512 field of view. That's not the optimal speed for this field of view. So I need to decrease the speed to 200 frames per second. So there is like a discussion between the number of the pulses and also how the pixel dwell time and how you're counting the photons and these kind of things. If you are interested, we are going to share this information with you as well, with this data. If you would like to read about that and find out what is the science behind that. But here, that's we find out that's with our sample, with our experiments, and how we are doing that 512 by 512 at 400 hertz imaging, 400 lines per second. we would get the pixel dwell time of 3.16 microsecond. That's quite okay. It works quite fine for us. Okay, so yeah, and here that's it's just about that's we would be able to change between the frame or the stacks. So which one? Now here that I would like to do the metabolic flame with imaging NADH and FAD, both of them in the salivary gland in the same sample. That's in the previous video. I have just find out the proper region of interest that I would like to image, and now I'm going to image these ones. Here we are. So first of all, that I need to add the second sequence for imaging the second one, because they have to image sequentially. So we first image the first one, so for example FAD, and then NADH, or vice versa. And for the FAD, the detection range, we set it from 500 to 550, and we use 900 nanometer laser line for the excitation. Just as a reminder, there is a solution and also a publication from Angelika Hook and Wolfgang Becker and their collaborators that's presenting a system, a dual beam infrared laser that you can simultaneously or super fast, you can switch between them. You can excite NADH and FAD and then imaging them simultaneously, so with two laser beam. Unfortunately, our fixed line is 1045, and we cannot use it to excite either of these two. So that's why we have to do sequential imaging. That's not ideal, but this is what we have, and that's our availability. Another solution was, as I mentioned another time, is Amase Priyasamy and his group. They have suggested to use single laser line and imaging with a specific detection range to almost avoid the contribution of the NADH signal inside the FAD. So that's like the bleed through them. But say here we are not doing that. We are doing the sequential imaging. So 900 nanometer for FAD and 417 nanometer excitation for the NADH. And we are using 6.25% laser power. That's what the value we found. It's working quite fine. And it's still 20 repetition. FAD is like that's more abundant than NADH. And in that case that we get really low signal, but what's happening is that now you see how long it takes to actually tune between one wavelength to another one. So I was adjusting with 900 nanometer, and now, so starting the 20 repetition for the NADH, 740 nanometer excitation, and the detection is 435 to 485. So it's taking 20 repetition and then accumulating everything on the lifetime image, fast FLIM image. And then we use this 20 repetition, the data from 20 repetition for the FLIM analysis. So when the 20 times for the NADH is finished, yes, here that we switch the second sequence, and it takes something like 16 seconds or about 16 seconds to actually tune to 900 nanometer. So we have a good question here as well regarding the beaming. Stop it here. Sandra is asking, beaming the pixel would help to increase the signal, but lowering the resolution. However, quite often the lifetime within a certain region of interest should be determined. Then the photons within the ROI are summed up, reducing the need for many photons per pixel and longer acquisition time. The photon number is mainly critical for pixel-wise beaming. Thank you very much, Sandra. I think that it was worth more than 5 cents. And yes, that's true. So we can do the binning. It can be done while we are acquiring the images, so using larger pixels. So I can choose a different zoom factor, and also that's different number of the pixels. So if I change the field of view now from 512 to 256 by 256, then the pixel size would be 2 times larger. So that would be equal to the binning of 2. Or in the analysis that I can use pixel binning and do the binning, and in that case that I would get better number of counts per pixel. That's exactly true. It might be necessary time to time to use these options. However, as I said, that we have done this mainly for the NADH analysis, and the FAD was the bonus, and we would like to see, we wanted to see that how the FLIR ratio looked like. That was really interesting for us as well. So continue with the acquisition. Now you see that when we get proper number of the counts in the field of view, that's the one indicator that actually if you might have proper count is that the decay histogram in the fast FLIM histogram would look like more confined. So you see that's for FAD, that's where we have, I'll just go back a little bit. For FAD, where we have less number of the counts, it's like that more broad. And also that for NADH, it looks more where we have almost proper number of the photons per field of view, which is about 8 million in this field of view. We have more defined histogram for the fast FLIM histogram. It's not exactly really like a, how to say that, it's not really an indication that you are absolutely okay, there is no problem. But this is one of the signs that you can see that if you're doing properly or not, or there is an option that you can play around with that. Okay, so I need to go back to the systems, to the microscope, and doing some analysis there. So just finishing the analysis that's started there, and then we go back to Connie very fast to talk about the results and what we have found, and concluding the workshop. See you soon. Okay, here we are. And share the screen. Okay, so say that we are going to do some analysis and just showcase that there isn't much to discuss here. This is the actual measurement that I showed the video of that, how it was recorded. What we do, we first of all, we need to discard the nuclei, and also that the parts that these parts, I have no idea what they are. Maybe they are also nuclei, but they look different. Maybe just in different plane they were, so it was like a roundish. It's a 3D sample, and this part as well. Maybe that there was kind of damage in the tissue. So I would just discard these parts. What I do is simply using the region of interest. That's what software is offering to us. And I'm doing that dirty and fast.
So all of them are included in the region of interest 1. As you see that I'm just choosing the nuclei and these two parts as well. And then when I say invert, what is going to be analyzed is only the cytosols and this part, which is the background. So here, I'm going to, first of all, fit all of them for the whole field of view, 2 components, and calculate an IRF, and I do the fit all. So, as you see, for the FAD, that since we don't have really proper counts, and there are some issues that with the number of the counts that we have, as well as the distribution of the intensity. So we have very, very bright pixels in some place, and also that we have very dim parts in some parts, that the fitting is not that great. Specifically with this sample that I have imaged, the fitting is not that perfect. Check the, oh, yes. Okay, I don't know how much you heard. I forgot this one, so here I don't have.
We could hear you, it was just more difficult to understand.
I see, should I repeat it?
No, that's okay. But from now on, we appreciate it, we have less background.
Okay, okay, great. So, but the data that we have for the NDD2, which is the NADH, it's quite very clear. So the chi-square, as I said, it's not 1. And my understanding from the chi-square from the software is that it's not reduced chi-square, so not necessarily should be 1. And this is what I am told. So 4, 5, it's quite okay. By the way, despite the fact that FAD image and also fitting that we get, it's not perfect, I would like to show you that's how the pixel by pixel fitting that is applied here. So the whole idea for the pixel-wise fitting is that you do the fitting in the region of interest in the field of view that you are interested in. So what we have done is the overall decay, the whole field of view, and then I have the RYs. So I'm interested to do the pixel by pixel on the RY, so I can, with the control, so like normal windows, I can just choose 2 region of interest that I have in 2 channels. And then I click on the FLIM image, and here, the whole idea is that when you do the overall fitting, you find out the lifetimes and you keep them fixed, you don't change them over the pixels, and then you try to find out the amplitude. So what is important here is that, as I said, when you are trying to do solving a problem using iterative algorithms for fitting, you need to make sure that you are fixing as much as possible the parameters that you can. Anyhow, for this case, I'm just going to define this area as background, so including everything else for the NDD. So the background would be something like here, few, something like 5, 6. I would say that, yeah, the rest is background. And fitting everything. I know that this is not the ideal one, but I'm going to fit everything here with the precise fit. And for further analysis, what we did was that just going to the free and extracting the parameters that we need, which was amplitude 1 from each of them. And here amplitude 1 as well. And going to the FLIM down here, save image. What is important is that the software is actually saving these images as 16-bit image. So there cannot be any digit there. So all the values in an image is integer. So
if I'm going to convert it without any factor or range, I'm going to end up with an image which is ranging from 0, 1, 2, 3, or something like that. So that's why I apply a factor or range of 0 to 0.001. It's actually divided by 1,000. I'm changing the values, so 1,000 times more. So that's how the gray value would look like. It's interpreted as the amplitude, or for the lifetimes when we have the nanosecond. So that would be translated to picosecond. So that would be for one of them, just giving that a name, and that would be test, for example, hands on A1. That was amplitude 1. And when I change the name, I need to make sure that I'm changing this value as well. As well as for the, so what I interested as well, that's amplitude 2. And I need to export the lifetime, the mean lifetime amplitude weighted as well for further analysis. So the further analysis, that's what we did. I'm just going to export this value as well, and this one as well. And saving them. So that would be TM, the same range factor. And what we have here are these TIFF images, which I can show you. And by changing the lookup table and so on, we can visualize them. So that's the mean lifetime for the first and the second one. And I can give lookup tables. So for example, black to cyan for one of them, and auto scaling. And this one would be black to cream. For instance, it's just a visualization, and that would be the overlap of these two. So they don't look really as fancy as they are shown and visualized on the FLIM module. So I just go back to the FLIM module and showing you how does that look like when we are at the FLIM module, because it's showing a nice overlay with the proper pixel resolution from the multi-photon imaging, intensity-based imaging overlay with the lifetime data at the top of that. And this is how typically it is visualized. On the free mode, I'm going to say that, okay, the brightness coming from the intensity, whole intensity, and the lookup table that I would like to have is the mean lifetime. And I would do the same thing, sum of intensity, and the lookup table would be the mean lifetime. And now I set the range to 2 second, and then this one, so that would be 3, and then this one would be something like 2. It's comparing 1 to 1 with each other, so I can put from 0 to 3 for both of them, and then comparing that one of them is more like reddish yellowish and the other one is only few. So I can make it a little bit brighter to see also the other ones. And we export this data and we use them for further analysis that has been done using R. It's also possible to export every single image, so A1, A2, and mean lifetime, and using Fiji for some analysis. So from here, actually, Mariana wanted to see something that with the phaser, just how does that look like? Here, how our phaser would actually look like. So we need to calibrate our phaser here, and I don't have the data for that. But the distribution is showing actually 2 of them, and then we can just remove this one. So I need to find the names, which one is which one.
Hi, Ali.
Yes.
Just wondering, why the harmonic is at 9? Can you decrease it by any chance?
Pardon?
The harmonic, you have below calibration, you have the harmonic. Can you put it in 1?
No, I can't. So here that might be the case that I can reduce it to 1.
Oh, yeah.
And yeah, so, but it needs calibration. That's a problem.
Oh, really? That's
yeah. So that's a point. And I said that it won't be really fancy because the phasor plot that on our system needs calibration. And yeah, so that's another reason we just focus on the FLIM for now. It's not only calibration with using, you know, I'm sure you know that. So you can calibrate the phasor plot using the exponential dye solution, and then you calibrate the phase and your plot. So here, if I load a mono-exponential decay, the data from a mono-exponential decaying fluorophore, then I can try to calibrate the phasor plots by finding out the phase and the modulation to adjust it to perfectly sitting on the semicircle, and then using that one for further analysis and further acquisition.
Yeah,
but
sure.
Our system, I have tried it several times, so I am in contact with the experts, so from the manufacturers, that how we can this one.
Yeah, it should be. Thanks.
Yeah, sure. So now I hand it over to Connie, who is going to present the data, and also that's explaining some of the parameters and also what we have found. Connie? Cornelia?
Yes, you have to
okay,
stop sharing
perfect.
the screen, otherwise I can't hop in. Yes, thanks. Okay, let's do. Okay, so since we only have a few minutes left, I will keep this relatively short and crunchy. So we ran with exactly the measurements that Ali had presented. So we used salivary glands as presented early, had a control treatment, or we treated with a glycolysis or an oxidative phosphorylation inhibitor. And what you see here is summarized measurements of 4 different samples each in duplicates, so in 2 different fields of view on the sample. You see the mean values represented as bar graphs, and what you can just kind of guide you through individual plots here. So the first one represents the mean NADPH lifetime, which I mentioned earlier is kind of a summary of a number of different parameters here, where you can see already there's a response of both, or at least a prominent response of the 2DG treatment of the inhibition of glycolysis on the mean NADPH lifetime. The response of antimycin A is relatively little. If we only focus now on the percentage of the short lifetime, so this being the free NADH, we can see that inhibition of glycolysis in orange reduces this short lifetime fraction, while antimycin A inhibition of oxidative phosphorylation increases this. So this was the tendency which we exactly expected in the beginning. Inhibition of glycolysis shifts towards more bound or long lifetime fractions, while antimycin A shifts towards glycolysis. Looking at the individual short and long lifetimes per se, we can see the short lifetimes are in the range of 0.5 nanoseconds, varying relatively little, while the long lifetime fractions are in the range of 3.5 nanoseconds. And you can also see that these differ between the individual treatments, possibly representing interactions with different proteins and enzymes up on these different it's a biochemical states of the tissues. If we go now for the data of FAD, we can see that also FAD lifetime, also the mean lifetime, differs between the different metabolic treatments. Looking at the short lifetime fraction, this increases in the case of inhibition of glycolysis, while it slightly increases in the case of antimycin A. So what you can see here is already that not in this case of the two inhibitor treatments, they don't respond in opposite directions relative to PBS, but they actually show the same tendency, but just to a different the same tendency, but just to a different extent. And now looking at individual lifetimes, you can see that also the short lifetime of FAD, being the protein-bound lifetime, the protein-bound component of a range of 0.34. So again, it's a bit higher than the published values or values that are known in the community. Upon treatment with antimycin A, this protein-bound fraction or the lifetime actually changes, which could also again indicate interaction with different proteins, while the long lifetime stays in the range of 2.6 to 2.8 nanoseconds. So this is just to summarize kind of one example of measurements one can do, or one can also use to kind of test the system, or also to get to know the sample or tissue you're planning to study and investigate. So again, showing you now the combination of NADH or NADPH and FAD as the FLIR ratio. Here you can see that it's in a range of 0.65, while upon inhibition of glycolysis, it increases slightly. Upon inhibition of oxidative phosphorylation, reduces, and this goes actually along with the interpretation from the Periasami lab, for example, that FLIR indicates kind of an extent of an oxidative state. Okay, so I will shortly summarize the biological interpretation of what we've seen, then leading over to Ali's final slides. So we use the technique of metabolic FLIM to detect in a label-free way metabolic states using autofluorescence of NADPH and also, if you like, FAD. We use two-photon excitation in the range of 740 nanometers and 900 nanometers respectively in order to penetrate deeper into the tissue, and it's also linked to less interaction of the tissue with the excitation and emission light, so in order to avoid phototoxicity. The FLIM redox ratio can be applied as kind of a lifetime redox fingerprint, as has been introduced by a number of labs now, and metabolic inhibitors inhibiting two individual pathways actually affect metabolism, and this is what we could show in this little experiment. And with this, I'd like to quickly hand over back to Ali. So I will stop sharing here if I can. Oh, I need to swap screens.
Okay.
Someone kicked me out.
Hello,
Okay,
Jane.
someone kicked me out. Thanks. Okay, good.
Yeah.
Thanks.
So, in summary, that's we have talked about the specimens, so NADPH or NADH, FAD are endogenous fluorophores in almost many cell lines, organs, and organisms. I'm not sure if it's in all, so that's why I mentioned in almost. So, meaning that in principle, that's labeled free. FLIM potentially can assess the cellular metabolism state in vitro, ex vivo, and in vivo. And also that's the instrumentation, as I mentioned that several times, beside the quality of specification of the instruments you are using, it is important that you know your system, you know that's the experiments you want to do with that system, and try to be consistent and optimal with the acquisition and analysis across the experiments you are doing that. But to understand the behavior of your system and also that how the algorithms working, you can use mono-exponential decaying fluorophores to study the response and also that how your system would actually behave and what are the algorithm analysis that actually respond to what you need. And the last one is analysis is try to optimize the number of the parameters to fit. So minimize the number of the parameters that you want to fit as much as possible, because when you increase the number of the components, so you increase also the number of the amplitudes you are going to fit. And then if you fit also the IRF shift and the background and so on, you have something like 10 parameters variable which ends up to enormous number of degrees of freedom statistically. And in some points, it is not my saying, somebody said it, you can feed an elephant with a multi-exponential decay algorithms. So that's important to have meaningful control measurements. So very last slide, I would like to thank Marco Brancak and the David Flores Benitez. The Marco lab provided us with the Drosophila samples, and David as well so supported us with preparing some of the samples. That's especially the ones that I use for recording the videos. And Magdalena Conchares, that's our PR, the CMCB in TU Dresden. She actually recorded all the videos that we had and also most of the images that we had from the system, and as well as edited them. And labs of James Sands and Nils Kreuger from BigCube, Simon Alberti, Dennis Gorbal, Stefan Grill, all of them from Biotech that have supported us and provided us with the chemicals and the lab space and instruments that we needed for conducting these experiments. And more importantly, I should mention our team at the CMCB, especially last few months that I was super busy with the organization. They have taken over some of the tasks that I should do routinely. And for sure, that's now, Connie has joined us from September, our FLIM specialist in the lab. So thank you very much. I wish you all a nice evening, morning, night, depends where you are living in the globe. Thank you.
Thank you all for hanging in.
With us.
And I'm going to answer one question, the last one regarding mean lifetimes. When should one use intensity weighted or amplitude weighted mean in lifetime? I don't know. Yeah, to be honest, the ones that I'm using is always amplitude weighted, but why it is said that it's more robust and reliable to interpret the data, I don't know. So that might be a good thing to address. That's tomorrow on the workshops with the in-depth analysis as well. Okay, Mariana has mentioned that intensity weighted is the one that people should use in TCSBC. Okay. Good. And Amazi's mentioned that always amplitude weighted is correct and reproducible. So that's what I see in our system, is that amplitude weighted is like reproducible and more robust. And that's true. That's for the FRET, as specifically it's mentioned that we have to use amplitude weighted. But what is the reason? To be honest, I really don't know. It's just that's how we are conducting the experiments here. Okay, if there is no other question, Stefan? So
don't think so.
To have any announcement, anything, so we can...
I just hope to see many of you back.
Okay, thank you very much.
Thank you, bye.
Bye.
Bye. Take care.